So, I guess it's reasonable to start now, Hannah.
Yep, sounds good.
It's 2 minutes past 2, and welcome to another meeting of the ReMicS network, the regional network on microbiota science. I will be, and thank you for attending amid everything. I'm not sure where you are, all of you. I'm at least at home. Some may even be at work. The lucky ones, I guess. We have hopefully what will be an interesting program today. I will talk together with Peter Hov and Johnsen, who will, I think, probably dominate the talk on recent efforts in our hospital, at Oslo University Hospital, to establish a fecal microbiota transplant system with a donor bank. And this is linked to a strategic research area for personalized microbiota therapy. You will see Peter as we move along when he starts to talk as well. I guess you can start, you can try to stop me if you want to ask questions while we talk, or you can try to say something in the chat and I can see if I'm able to follow up on it. Or we can get help from Hanne if she is available. Okay. This is then the topic, and the next slide is actually more or less like a throwback to something I had an internal seminar talk a couple of years ago, and then it was related to new funding to research on clinical microbiota medicine, which you are all a part of as part of this network. And one thing is the ReMicS network, which was the first point, but which was initiated in 2019, but at the same time, we got some internal funding in Oslo University Hospital with a bit more focused title called Personalized Microbiota Therapy in Clinical Medicine. So there has been some resources that we have with more or less the same aims and topic, I would say. But I would talk a bit about it, and then we'll focus on the FMT-related topics. Just a brief comment on what strategic research area is, and it's a small, not very well funded, I would say, but small system within the hospital from the research section, the division, where they want to stimulate some specific areas. So now and then they had a possibility to apply for this funding. And the idea is that it should focus on some kind of multidisciplinarity within the hospital. It should be relevant for the patients, and it should definitely strengthen existing infrastructures, and also somehow, if it's possible, try to stimulate the activities and careers of younger scientists. And right now there are 4 of these strategic areas ongoing, and there will be new ones, I think. And one of them is, we know very well, is the TTA, the turning the tide of microbial resistance. And then you have some other areas. One is related to ex vivo drug testing in colorectal cancer, and one is on cell-based immunotherapy. And then it's our area, which is then personalized clinical gut microbiota therapy, more or less. And part of this personalized gut microbiota therapy in clinical medicine, what does it mean, and what's the aims here? This means treat the gut microbiomes to perform this therapy, of course. And then the next step, of course, is also, or the separate step is to look at the biomarker aspects, how to use gut microbiota profiles, for instance, and how that could potentially guide clinical treatment. And the ultimate goal to personalize this, that we are able to not go all to target specific therapies to specific individuals based on some biomarkers that may be microbial. And of course, the key idea is to give opportunities to young, talented researchers, etc. And this is how it could be seen. You probably seen some things like this previously. This is from a commentary several years ago from Sharon Ras. And he speculated that we could potentially have clinical microbiome-based diagnostics. We could hopefully personalized select treatment based on this microbiome readout. So it could either be for targeted microbiome modulation, but of course it could also be that you could select treatments, other types of treatments based on the microbiome readout if the microbiome actually predicts the effect of any common drug. And then it's of course the actual modification of the microbiome, which could be precision probiotics, prebiotics, drugs, or FMT. That could hardly be considered precision. And then you could potentially even think of monitoring treatment success. So some examples. And the structure of this strategic research area is very similar. If you ever saw the application for the ReMicS network, it's very similar. But we sort of try to define some basic skills of core specialists. We think gastroenterology interested people, some people who know who are specialists on infectious diseases, and we need. I get the idea, the comment that they have an unstable internet, and it may of course be that my children are gaming. So let's immediately go for. Is it possible? No. I can turn off my video, of course. Let me know if it's still a problem. In addition to gastroenterology, infectious diseases, and microbiology, we do need, of course, the specialists of the diseases that we're interested in. So we have sort of like a set of basic skills that we need to have related to these endoscopy, microbiology, and of course a donor bank. And then we need to solve these clinical problems within the hospital and through our patients' problems that we try to solve. And the idea is really to make microbiota medicine relevant and make it a research topic relevant in many groups. So not only us that have this as our main interest, but also a majority, I would say, of diseases where this should be investigated. And just to remind you how the ReMicS network is organized, here we are talking a bit more about basic skills and clinical skills. But more or less it was the same concept. So these are parallel structures, I would say, although it's a bit more focused in the strategic research area. So what is in such a grant? It's not much, but it's some funding, which is valuable. 1.2 million kroner every year for 6 years. So some long-term aspects here. I think we defined it that it was possible to hire one person 100% and one 20%. So far, it's a bit more flexible how we have done this, but we will see as it goes along, and there will be some funding for running costs. So the main aim is really to develop projects and to focus on this donor biobank and therapy, which I think is our key tools that we need to make personalized clinical microbiota medicine a reality. But formally, it's part actually of this Norwegian PSC research center, this liver disease that I'm interested in, which is then a part of the Department of Transplantation Medicine. Also, a couple of years ago, we had some ideas on where we would be in about 5 years, and fairly bold aims, I would say, that gut microbiota composition would influence choice of treatment in more than one condition, and also that more than one condition would be treated with personalized gut microbiota therapy. This is a tough one, I must admit. While the third point here, is there a need for a microbiota unit in the hospital? And I think we've already concluded that we need this, and it's more or less been developed already, as we'll come back to. So this is, I guess you know, all of you, what is fecal microbiota transplantation? You have simple figures. It can be prepared in many ways. You could try to make it more popular using capsules. These are now usually colored, and it's quite commonly used in trials here and there. I'm not sure there are too many capsule producers in Norway yet. What's the relevant evidence? Why do we need this? This is probably also somewhat familiar to you. I think the strongest evidence, or that's what really brought this to common clinical practice, was the New England Journal paper on FMT for Clostriditis difficile colitis. It's now established practice for recurrent disease. I think when going back to this paper, I always get astonished looking at the numbers here. 16 FMT treated and 13 vancomycin treated. Of course, the cure proportion is very high. It's very convincing data, and it's also based on a long history of practice in this field. But the numbers are definitely small for a New England Journal of Medicine paper. And then you have relevant evidence in IBD. There are several studies that have showed some promise. This is, I think, was from Australia, where they had a very intense treatment strategy with endoscopic regular enemas to treat ulcerative colitis with FMT. And as you can see, there is actually an interesting proportion that are in remission or have a response, but a very low placebo rate, which is, I think, it's fascinating how low the placebo rate is, which is not common, I would say, in clinical conditions. But several studies with the positive results suggesting that there is something, at least here, that is promising. And then you have, I would say, other non-GI conditions where you really have, there is a huge interest and hopes, but it's difficult to know where it will end. But I think it's fascinating still. It's going back already to 2012, where they from Amsterdam published the fat loss trial, where they gave full fecal transplants from lean donors to obese donors. They didn't have any effect on the weight, but they measured insulin sensitivity, which definitely increased from before to after. I think you see the range here is fairly wide. So it was a marginal result, but it's fascinating to see that you could see actually an improvement in the glucose regulation just by fecal transplant at 6 weeks. And then they repeated this and was published a few years ago. And it was actually exactly the same result. You have an increase in insulin sensitivity after 6 weeks when you get an FMT from lean donor. So very convincing that this is in fact true. But the effect was lost at 18 weeks. So sometimes between 6 weeks and 18 weeks, the effect was lost. So here we really have just lots of opportunities and many great, or at least many ideas on what we can do, but we need the tools. And at least in Oslo, we have not had the tools. We have just had this longing for a donor biobank. So we have done this big question, how to make a donor biobank for fecal microbiota transplantation. And I guess our answer then was to hire an external expert. And this is where you are involved, Peter. Peter?
Yes, thank you, Johannes. I take it from here.
Yes, so just for everyone, we hired then Peter in the strategic research area in a 20% position, really to build this up. And you can perhaps present yourself and what you and we have been working on.
Yes, you can just go to the next slide, please. Okay, thank you. I will in brief tell about my background. I have worked as a physician at the Department of Internal Medicine at the University Hospital of North Norway. I finished my PhD on a single center study testing the efficiency of FMT in irritable bowel syndrome in April last year. And I have established a stool bank at the University Hospital of North Norway in Harstad. Currently, I'm involved in 5 different RCTs testing the efficiency of FMT in morbidly obesity, chronic fatigue syndrome, irritable bowel syndrome in a national multicenter study, primary Clostridium difficile infection, and in another study that currently is under planning. Next slide, please. Okay, so a cornerstone paper from 2015 said that the microbiota in host health is a new clinical frontier. Looking at the studies that are now being undertaken, it seems like the forecast in this paper has a good chance of being proved correct. Next slide, please. In a search on clinicaltrials.org in January 2020, approximately 1 year ago, I found 195 studies testing FMT as a therapy in clinical trials. I repeated the search this week and found out that there are now registered 412 studies with FMT as a clinical therapy. If personalized microbiota therapy is a strategic research area, then a stool bank is needed to compete in this race. FMT is being tested in autoimmune disease, in neurodegenerative disease, such as Parkinson and multiple sclerosis, in psychological disorders, in functional disorders, such as IBS and chronic fatigue syndrome, in metabolic disorders like Johannes showed, and in morbidly obesity, in cancer treatment to see if FMT can work in synergy with immunomodulating treatment to increase the tumor suppressive effect, in hematological disease to decrease graft versus host after allogenic stem cell transplantation, and in infectious medicine to eradicate carriership of multi-resistant bugs. In common for all the examples of FMT as a clinical therapy is that there already exist indications of a clinical significant effect through data from pilot studies. Thus, the stool bank may be of importance for many different disciplines and focus areas at the University Hospital of Oslo. Next slide, please. So, a stool bank is needed for further development of FMT as a clinical therapy. Several signaling pathways for interaction between the gut microbiota, the immune response, and the central nervous system has been described. However, they are not fully characterized, and I think more pathways are likely to be discovered. A better understanding of the signaling pathways between the microbiota and the host is needed to develop personalized microbiota treatment. A better understanding may also lead to the development of other therapeutic agents aimed at the pathological signaling pathways where the microbiota is involved. Therefore, I think that a stool bank in Oslo in synergy with omic platforms will lead to more research and a better understanding of the role of the microbiota in host health and disease. Next slide, please. For my PhD, I did a double-blind randomized placebo-controlled single-center study testing the efficiency of FMT in irritable bowel syndrome, hereafter referred to as IBS. IBS is a disorder characterized by abdominal pain in relation to alternations in bowel habits. However, extraintestinal complaints like fatigue, joint pain, and brain fog are also common. In this study, 90 participants were assigned donor FMT or placebo. Placebo was FMT made from the participants' own stool. 3 months after treatment, we found a significant effect of donor FMT on bowel-related complaints. Next slide, please. We also found out that fatigue was responsive to FMT treatment in the subgroup of participants with IBS and self-reported depression before treatment. Interestingly, the subgroup with IBS and depression had a fatigue score almost twice as high as the other subgroup with IBS and no self-reported depression. Since fatigue is a symptom that is a common complaint in many other disorders, I think this is an interesting finding. Next slide, please. The stool bank at the University Hospital in Harstad is developed in synergy with the IBS study and other ongoing clinical trials using FMT as treatment. Neither studies could have been launched without the production of FMT products organized in a stool bank. Over the last 3 years, the stool bank has made approximately 600 FMT products. Most of the FMT products are made for use in clinical trials situated at the University Hospital of North Norway, but also for the colonized study with FMT for primary infection with Clostridium difficile, performed by the Clinical Effectiveness Research Group at Oslo University Hospital. Next, please. The development of the stool bank in Oslo is done in a collaboration between the Norwegian PSC Center, the Clinical Effectiveness Research Group, the initiative with personalized microbiota therapy as a strategic research area, which finance my position, and experts in microbiology and infectious disease at Oslo University Hospital. Without the effort Fredrik Emil Juel and the Clinical Effectiveness Research Group has put into this project, the realization of the stool bank would have been very difficult. Siv Isaksen, research nurse at the Clinical Effectiveness Research Group, has also put in a lot of effort to establish the infrastructure needed for the stool bank. Next, please. The most important aspects of the quality and safety control of the FMT products from the stool bank includes recruitment and screening of donors, processing, traceability, and quarantine of FMT products. I will in brief talk about these aspects. The quality and safety control is based on the last consensus report from leading international experts in the field of stool banking. Next, please. For the stool bank, we want healthy volunteers, 18 to 30 years old, and plan to invite students at the University of Oslo that through the studies frequently are situated at the hospital. They are paid 250 Norwegian kroner for each 60 grams of stool delivered, with a maximum payment for each stool donation of 1,000 Norwegian kroner. At the stool bank here in North Norway, we experienced that some donors made more money from each stool donation than what could be justified by the effort. And that is why we have established a roof of 1,000 Norwegian kroner for each donation. There may be studies that want donors for personalized FMT treatment and want to select donors by criteria that differs from our standardized donor FMT protocol. If so, it is possible to adapt the screening process based on study-specific donor protocols from other researchers. Next, please. We start the donor screening with a questionnaire to determine if donors are completely healthy and to ensure that they do not engage in any behavior associated with increased risk of transmittable disease. With completely healthy, we mean no use of medication, no previous history with autoimmune disease or digestive disorders, and no complaints associated with any functional or somatic disorder. We also ensure that there are no signs or risk factors associated with disorders where an altered gut microbiota is found. This is by far the stage in the screening process where the largest share of potential donors are excluded. Next slide, please. After the questionnaire, donors are screened with a standardized panel that includes blood, stool, and urine samples. The screening is mainly to reduce the risk for transmittable infectious disease, but we also obtain general biochemical parameters to reveal any undiagnosed active disease. The screening for infectious disease is based on the current leading European consensus report, but adapted to Norwegian conditions through the work of an advisory group that include Jørgen Bjørnholt, Christian Thunby, Johannes Hov, Fredrik Emil Juel, and me. Next, please. I've heard about one happening with delivery of donor stool covered in toilet paper. This was from a donor that delivered from home, and it turns out that the stool was pulled out of the water in the toilet at home. So to ensure sanitary conditions, we want the stool delivered from donors using a portable toilet chair situated at the hospital. Sorry. Okay. This also limits the time from delivery of stool until processing into a FMT product. Since exposure to ambient air is shown to reduce the viability of the microbiota in the FMT product, this is also a safety measure that increases the quality of the FMT product. Next slide, please. One FMT product contains 60 grams of donor stool homogenized in 120 milliliters of saline and 25 milliliters of 85% glycerol. So if donors deliver 600 grams of stool, we can make 10 FMT products from one donation. Donations with 600 grams of stool are rare, but it has actually happened, and this is like Christmas Eve for the stool bank. Next. The first picture show us the equipment needed to make one FMT product. The filter is custom made for stool banking. Most of the equipment is certified for medical use and as a minimum certified for use in food production. The donor stool is homogenized with a blender before filtering and drawing on the 50 milliliter Lurlock syringes. We draw the fecal matter using a technique that empties the syringes for room air. The processing procedure is easy to perform and minimize the exposure to aerobe conditions as much as possible without using anaerobe chambers. After processing, the FMT products are stored at -80 degrees. Next, please. From each stool donation, we collect 3 5 milliliter Eppendorf tubes with donor stool. The tubes are labeled with a code that corresponds to the FMT product code. The traceability is an important safety measure. If any adverse events occur after a FMT that may be related to transmittable disease, we can test if it's caused by pathogens in the donor stool. The aliquots of donor stool on Eppendorf tubes also allows for use of omic platforms to determine if changes in the recipient of the FMT product mirrors the donor stool microbiome or metabolome. Next, please. In the study with FMT for IBS, biobanking with traceability allowed us to compare the microbiome to responders and non-responders before and after treatment with the corresponding donor stool. We did the biobanking of donor stool on aliquots like shown in the previous slide and used the same labeling system. Comparing responders and non-responders to donor FMT treatment, we found significant changes in the microbiome to responders associated with the metabolism of carbohydrates. This is a finding that links MAP and terrain, as patients with IBS often find relief from a diet that limits the intake of certain short-chain carbohydrates. We also found that the microbiome in responders change more in the direction of the donor microbiome than what it did in non-responders, and that the changes lasted until 12 months after treatment. The 12 months follow-up was the last time point for biobanking in the study. The study was small with 14 responders and 8 non-responders to donor FMT product treatment, and they are therefore not able to draw any firm conclusions. However, it shows that one treatment with a FMT product can induce long-term changes in the recipient's microbiome, and that we, through sequencing of the microbiota, can pinpoint these changes and use them to reveal pathophysiological disease mechanisms. If you want to know more about the sequencing analysis performed in this study, I suggest you talk to Rasmus Goll at the University Hospital of North Norway, the main author of this study. Next, please. Okay, now back to the safety and quality control. FMT products are stored in quarantine until a pre and post screen are performed. We screen the donors in cycles of 8 weeks. Each cycle includes the donor screening questionnaire I showed a few slides ago, and a standardized panel with blood, stool, and urine tests. In addition, donors have to answer a short questionnaire for each stool donation to ensure that they are not carriers of transmittable disease intermittent between the complete screening cycle every 8 weeks. Screening for COVID-19 and with the throat swab and serology is performed every 4th week. FMT products processed in between one screening cycle of 8 weeks is released from quarantine if there are no positive findings from the pre and post screen in the screening cycle where the donor stool was obtained. The figure to the right shows how stool for FMT products is obtained in each cycle and how FMT products are placed in quarantine until the screening cycle is complete. Next, please. This figure shows how one positive finding leads to discarding of all the FMT products in the screening cycle with the positive finding. Next, please. So this is mainly the most important aspects of important steps in the quality and safety control of the FMT products in the stool bank. I hope the establishment of the stool bank will lead to more studies with FMT as a clinical therapy and more research in the role of the gut microbiome in health and disease. Thank you very much for the attention.
Thank you, Peter. I have a few things to the end here because we have really quite nicely gone through how such a donor bank should be established and for optimal safe procedures, etc. But there are also other challenges in this field. It's like that we have not really gone into, like, what is an FMT, what kind of regulations should it relate to? We know that we also talked about it in our national meetings, that FMT in some countries has been defined as a drug, for instance. While I would say we definitely think that it's not very suitable to call it a drug. And it would make a lot more sense to call it, to look at it as like a tissue, more like a blood product or as a transplant, actually. And this is what we have done now. And actually what has taken place is that in the Department of Transplantation Medicine, where this strategic area is organized, the department head, Paul Dagliner, is a transplant surgeon. He has kind of established an FMT unit and informed, so it's more or less accepted in the division that we now have an FMT unit. And in this establishing of this, the department head states that we consider this as a transfer of cells and tissue, and will handle it according to those regulations. So this is more or less, this is what we have determined, and we can then work on from there with the support from our department head. And it makes lots of sense, and that it's actually a part of the Department of Transplantation Medicine. And the use of the donor bank will be initially mainly scientific clinical studies, but of course we aim to make this also a source for what we think will be increasing clinical activity. Also because actually the clinical activity already ongoing, which is so far is now and then a recurrent C. diff treatment, need proper donor products. And with such a bank, we can provide those. So this is really what happened now, and based on the work that has been done. I think also we should mention that there is actually a Nordic Trial Alliance in fecal microbiota transplantation, which I think initiated from Aarhus, Christian Vaz, who was talking at our national meeting 1.5 years ago, I guess it was. And we have a representative from Oslo, Kjetil Garberg, at gastroenterology at Rikshospitalet. Where we're aiming really to collaborate on methods, regulations, of course, trials. And also, I think importantly, establishing formal structures for like a peer audit of FMT centers to make sure that these are running with acceptable practice. Because as we extend indications, we do need to make sure that we do it at least according to guidelines and as safe as possible, since extending into, I would say, non-life-threatening conditions, we will really need to make it properly. This is also something that we are involved in. And of course, the donor bank is just the beginning. If you want to personalize it, we need a larger selection of donors that we can profile, so we can select the right donors. And of course, as Peter also alluded to, we have the opportunity of collecting special donors only for research purposes. It could be, for instance, lean donors for obese people, if that is something that would be of interest to us. So to conclude here, we have a strategic research area in the Oslo University Hospital called Personalized Gut Microbiota Therapy in Clinical Medicine. And it's funded for 3 to 4 more years. And we have now established a donor bank for FMT. The first donor has formally not yet been included, but everything is more or less up and running. I would say a key point in the talk and in general with this practice is we need optimal strategies for quality and safety control. And of course, this will now hopefully give us a lot of opportunities ahead. And I guess that was the last slide. Thank you very much for listening. We're happy to answer questions either with a chat or just raise your voice or your hand. Okay, we have a question here from Siv Bøhn. Is the frozen and thawed FMT comparable to that of fresh FMT in terms of clinical efficacy? In other words, does the freezing process affect some bacteria more than others? I guess, Peter, you did in part investigate this in your IBS trial?
In the IBS trial, there were no significant difference between the effect of fresh and frozen donor FMT. And I think the same is found in Clostridium, and that freezing influences the viability of the FMT product, or the viability of the microbiota in the FMT product. But as far as I know, exposure to ambient air is a bigger threat than actually the freezing process.
I think it's also important to say here that fresh FMT, as you already saw from the quality and safety concept, is not really an option. Okay. We do need to freeze it, and we need to close it with our post donation screening before it's possible to give it to anyone in a safe way. So that would, if so, you would really need a special protocol to really go for fresh donations. Other questions?
I might have a follow-up question to that. Do you see any challenge with, if you're talking about the air aspect, do you see a change in the distribution of the microbiota after the treatment? In respect, for example, for hyper anaerobic bacteria like the Firmicutes, they tend to be more prone to oxidation than or air than Bacteroidetes, for example.
I think that's a good question. I'm not sure the data you have from your IBS trial are enough really to say that there are particular groups of bacteria that are changed. So I must say I don't have the full overview of the literature here. It's, I guess, the donation and treatment protocol is optimized also for feasibility and based on experience that it has an effect. Of course, later on there may be we may need to adjust protocols if we don't get the desired effect. and we suspect that there are bugs in there that we would like to treat patients with, etc. But I think so far the major obstacle is just to get this up and going, more or less.
And one more question, that is, how is the complexity in numbers of bacteria when you have conducted the FMT treatment? Do you have any indications there that you have lower numbers of viable bacteria in the treated groups?
Do you know anything about this, Peter?
Well, you see, I'm not sure. You have seen increased diversity after FMT treatment, but sure that's the same as increased viability, so I'm not sure.
Very good. We still have some time.
I see Phil have a question.
Yes.
Yeah. Thanks, guys. Great, interesting talk. I'm going to ask a question, and I don't know enough background on FMT, but this is more curiosity. Do you know, or is it known that the effect from treatment is coming from kind of shift in microbial activity as a result of the transplant, or is it coming from a metabolite or a product that's come from the donor?
In recurrent Clostridium difficile infection, they did actually cure patients with sterilized fecal matter from filtering process before giving the FMTs.
So it's not necessarily a metabolism from a donor bug, it's more likely something that's shifting of the recipient, either from a metabolite or from...
This is, of course, an extremely interesting question. This sterile filtrate treatment was very interesting. For C. diff, it seems to be a bit of a particular situation, where there could be like inhibitory substances from the microbes that are still active after sterilization, after filtrating the actual bacteria, and that could inhibit the growth of the C. diff. For other conditions, it seems unlikely that you need some kind of take, that they actually are able to colonize to a certain degree the gut, so it's able to make an effect. But whether that is by removing the old one or by producing new metabolites, etc., is not known. You could, of course, also envision sort of a adjuvant treatment with, let's say, fibers or food for your preferred bugs, even to improve therapy. But this is just imaginations of the possibilities.
In the IBS study, we saw that a change of the microbiota towards the donor microbiota was associated with a positive treatment effect.
Was that taken with like 16S fingerprint, or was it looking more at kind of microbiome function?
Deep sequencing, and Rasmus Goll know more about that paper than.
Okay.
Yeah.
You could also already, when you think about personalization, there are all the treatment trials out there with what they would call a rational donor selection. For instance, what they would call butyrate producers, etc. To what degree that has been a success, I don't know really. It depends on the conditions. It's really widely different diseases that are treated, or where this treatment is tested. So, and you can definitely not expect an effect from everything. But if this is somehow a tool to identify where it's relevant, and then we can use it to identify or understand what's actually going on, then that would be the ultimate goal, I guess. Okay, perhaps if there are no further questions, I guess the meeting is about to end. There will be a new meeting. I don't remember the date.
April 14th, Knut Rudi will present for us?
Yes. Okay, so Knut Rudi at for April 14th, you can take a note and date in your calendars. Of course, it will be also circulated reminders when we are approaching this date.
And as
Okay.
always, we'd love to hear from you if you have any news to share. It's difficult to be very social right now, so please contact us if there's anything
Yes,
that you want to share.
Yeah, then we can distribute news or positions or whatever.
Yeah.
Very good. Thank you everyone for attending, and hope it was okay. And we will be back in April, and see you all then.
Bye,
Okay.
everybody.
Very good.
Bye.
Bye.